Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Steve Perry from Catalent Biologics.
Thank you, Leah. Thank you all for joining this webcast. I'll be speaking about enabling success in biologic drug substance development and biomanufacturing by leveraging expertise and collaboration. As Leah mentioned, my name is Steve Perry. I'm Senior Director of Manufacturing Sciences. I'm based in Bloomington, Indiana. Just quickly, introduction on Catalent. We're a global company of over 10,000 employees with over 80 years of experience partnering with pharmaceutical, biotech, and consumer health innovators. We improve the lives of patients worldwide. Catalent Biologics is that part of Catalent that focuses on biopharmaceutical contract development services. And we help our clients deliver life-saving drugs to patients quickly and reliably with advanced technologies and integrated service to support your product development needs from cell line through finished product supply. Just a few notes about some of our technologies at Catalent. Smart Tag technology is used in the development of antibody drug conjugates. Those are, which of course combine the specificity of monoclonal antibodies with the potency of chemotherapy agents and other payloads by conjugation with chemical linkers. Smart Tag technology enables better control of site-specific conjugation, improving potency, safety, and stability. Our GPEx cell line development technology generates high-performance, highly stable production cell lines in a wide variety of mammalian host cells. To date, over 500 products have been produced using the GPEx system, with over 50 lines in clinical trials and over 10 commercially approved products. Catalent has 2 state-of-the-art drug substance manufacturing facilities using both single-use and stainless steel bioreactors. Our Madison, Wisconsin facility was recently expanded and features single-use bioreactors up to 2,000 liters. Our Bloomington, Indiana facility has stainless steel reactors up to 2,500 liters with smaller single-use reactors and won an ISPE Facility of the Year award. At both sites, both Madison and Bloomington, process and analytical development services support manufacturing operations. Catalent performs drug product manufacturing in prefilled syringes in Bloomington and Brussels, Belgium. The Bloomington facility also has capability for filling of vials, that's liquid or lyophilized, and cartridges, as well as packaging. The Bloomington facility also received an ISPE award for our innovative flexible filling line. And of course, drug product manufacturing is also supported by formulation and analytical development. In addition, Catalent operates 2 state-of-the-art facilities offering comprehensive analytical services, including protein characterization and bioassays. Services include method development and transfer, as well as in-process release and stability testing. And then, of course, all these services are provided under the Catalent Biologics umbrella, ensuring seamless solutions from development to packaging. Let's get into a discussion on biomanufacturing here. Process development really consists of 4 main areas that must be completed before we can go into GMP manufacturing. You see them here on the slide here: cell line development, upstream, downstream, and analytical development. Now, you can imagine, in principle, it would be nice if these activities could occur in sequence. That is, the cell line must be developed before upstream development can occur. It would be nice if the upstream process were finalized before the downstream process was developed. And it would be nice if the downstream process was finalized before analytical methods could be finalized. Of course, in reality, that's not the case. Timelines won't permit it. And so we have to be smart about when we have enough information to proceed to the next stage, even as we're finalizing the information for the previous stage. So at Catalent, we've worked with a large number of projects and clients, and we can recommend strategies to accelerate the development timeline. One other note, while Catalent has the capability to perform all of these development functions shown on the slide, many projects come to us from clients who have already completed some of this work. In fact, in some cases, the client has acquired the product from a third party and may not have direct access to the developers. In all these cases, Catalent can work with the client to quickly get up to speed based on whatever information is available at the time, so we don't unnecessarily duplicate work that's already been done. This slide shows a bit more detail on the upstream development. For early scouting of bioreactor conditions, we use small-scale shake flask studies as well as the AMBER bioreactor system to rapidly screen conditions. The extent of process optimization will depend on the timeline. For well-behaved monoclonal antibodies, we have platform processes that we can use as a starting point. Another objective of upstream development is to provide material for downstream studies. Moving on to downstream development, it's discussed on this slide here. Scale-down models are used for chromatography and filtration steps. Again, as for upstream development, platform processes are available as a starting point if the client is not transferring in an already established process. The development effort may include generation of tox and reference standard material. An engineering batch may be done prior to GMP manufacturing, or it may be waived if the process is considered straightforward and the molecule is well behaved. I wanted to speak a little bit about the difference between early phase and late phase development. And the scope of the development effort will really depend on the clinical phase of the molecule. For preclinical and phase 1 or early phase, the focus is on getting a process in place quickly to provide material for clinical trials. At this point, it's not really economical to perform extensive optimization or process characterization. For example, for monoclonal antibodies, we would apply a 2 or 3 column platform purification process along with a platform analytical package for antibodies. Looking ahead to drug product development, we would perform a high throughput formulation screen, mixing and shear studies in support of manufacturing, and process container closure material compatibility, and if necessary, lyophilization cycle development. For late phase development, the molecule has already shown promise in early clinical trials, so additional development work is justified. Process characterization begins with a risk assessment of process parameters. The design space is identified through DOE and other strategies. And then finally, a control strategy is developed linking quality attributes to process parameters. We could define parameters as critical or non-critical at this stage. And the analytical strategy is defined to address method capability and attribute detectability. Extended analytical product characterization is performed. And again, looking forward to drug product formulation and lyophilization cycle is optimized, and the drug product process is characterized. Once the process is developed, it's transferred to the GMP manufacturing facility. This slide shows some of the things that must be considered during tech transfer. An important element is facility fit, meaning an assessment of the manufacturing facility to execute the scaled-up process. Now, for processes developed internally, obviously at Catalent, it's less of an issue because the developers, of course, are already familiar with the manufacturing equipment and will work with the manufacturing scientists and engineers to make sure the process will fit. For processes transferred in from clients, a detailed assessment must be made. A process will be sketched out in our facility at the desired scale based on data provided by the client. The process will then be scaled down and assessed in process development. In some cases, targeted development may be needed to adapt the process to our facility. This is really typical with any CMO, as equipment will always vary between facilities. Tech transfer is an interdisciplinary process, primarily involving process development, manufacturing sciences, process engineering, manufacturing, and of course the client. The initial outputs of this process are the upstream and downstream process descriptions, which are technical documents describing how the process will be executed at large scale based on the small-scale development data and based on engineering scale-up principles. The process descriptions are used as the basis for GMP documents, including master batch records, which are reviewed and approved by the quality organization and will be used by the manufacturing operators to execute the process. For process validation campaigns, additional work is necessary. This includes the process characterization mentioned earlier. This activity establishes the acceptable ranges of critical process parameters based on critical quality attributes of the product. Quality risk assessments are performed to ensure that the manufacturing process will control these parameters within an acceptable level of risk. If necessary, mitigation measures are put in place to reduce the risk, and the master batch records are updated. In addition to the main process performance qualification protocol, related studies are performed as part of the overall process characterization, or as part of the overall process validation package, and you can see some of the studies are shown here on the right of the slide. In the next few slides, I'd like to touch on a few points that are key to ensuring a successful collaboration with the CDMO. Project success depends on a shared understanding of objectives. You can think about in any endeavor, not just biomanufacturing. There's always a potential inherent tension between quality, cost, and speed. Critical quality attributes are typically fixed and driven by clinical requirements. For biosimilars, they may be particularly rigorous because of the additional need to match the attributes of the innovator molecule. So, of course, quality comes first and is really set. Once those quality requirements are set, then you look at objectives of cost and time. And typically, in most of our projects, the available time is what defines the development strategy. So a process will be developed within that time that will deliver a certain titer and yield. If additional time is available, more development work may be possible to increase the titer and or the yield. Another consideration is the intended scale of the process. If clinical supply requirements can be met at an intermediate scale, it may be possible to shorten the development timeline by deferring consideration of issues that will be significant at a larger scale. So for one example might be harvest. It might be possible to shorten the development timeline by implementing an initial filtration strategy that may be feasible at intermediate scale, but maybe not practical at large scale due to filter area requirements. So these are some things that can be done to shorten the timeline, but it is very important that conversations take place between the client and the CDMO so that all parties understand the trade-offs, particularly circumstances change later on, as they often do. It's also important for the client to understand the resources that will be required and the timing of when they'll be required. At a minimum, a client technical and quality representative will be required. The client will also need a project management contact, which would be the person empowered to make decisions on behalf of the client's company. At smaller companies, this could be the same as one of the technical or quality contacts. That's certainly possible. It's also a good practice to define an escalation pathway to quickly resolve issues that cannot be solved as a project by the project team. It's always good to have that defined ahead of time. I want to talk a little bit more about client resources and collaboration. The extent and role of the client resources will vary by the client company and by client company size and also the type of project. So we can look at two typical examples here. For example, if the project is a tech transfer from a large pharma, in other words, the large pharma already has the process and is transferring to Catalent, then in that case, the client is a process expert, likely has a number of different experts on the process who are very well experienced with that process. In that case, Catalent, the subject matter experts, are the recipient of the process knowledge. And of course, we'll rapidly develop expertise in the client's process as we become more familiar with the process in our own hands, and also through our general experience with biomanufacturing processes. You can look on the other hand, if the process is developed by Catalent and the client's a small biotech company, maybe with very limited resources, then really Catalent would be the process expert, and we would transfer the process knowledge to the client in the course of the project. In addition, the involvement of client technical resource will vary based on client preference. Some clients are interested in very detailed involvement in the development discussions and decisions, so they like to really be very well involved in those talks and those meetings. Other clients just prefer to provide higher level, less detailed oversight. Either way works for us. Catalent has worked successfully under both scenarios. In fact, we enjoy the interaction and the scientific discussions that take place. But really, it just depends on the client's preference in terms of how much the client prefers to be involved in these discussions. But the client should consider at the beginning of the project what level of involvement they would prefer or to be able to support. And I would say in all scenarios, again, the relationship is most productive if the spirit of scientific collaboration is maintained. Third is that it's important the client understand resources needed for document review and approval. So sometimes this hits the client by surprise, and we try to make sure the client's aware of these resources that are required. They will become more significant as the process is transferred from process development into GMP manufacturing. In particular, there will be greater involvement of the client quality representative. Catalent will provide a batch record review schedule and tracking as documents proceed through review and approval. We've got some pretty detailed tracking tools that we use for that. And so, again, it's important that the client understand and that we have agreement on client review and approval details, such as number of reviews, timeline required for review. Important one is backup personnel in case people are on vacation and so forth. Those things should be established early on to have a smooth process of document review and approval. And finally, talking about oversight of the actual manufacturing campaign. As a CDMO, of course, Catalent is responsible for the campaign, overseeing the manufacturing campaign and providing frequent updates to the client. Typically, we'll provide an upstream update will be during passage days, and then every day through the production bioreactor, and then every day through downstream up to bulk fill. The client may elect to be present on site during manufacturing. That's certainly done and certainly possible. And in fact, that's governed by the quality agreement and the project plan. In case of unforeseen events, rare, but they do occur, Catalent will respond and will recommend a course of action. It may be necessary for the client quality representative to provide concurrence, which may even be in real time in case there is some action that has to be taken. So again, that should be planned for, and there should be an awareness that those resources may be required per the quality agreement. Just a couple other considerations before we wrap up here. A couple things to be aware of are sampling and testing plans and materials. These are some things that might be considered earlier. It's better to consider them earlier rather than later. Sampling plans, that includes all the testing, including release testing, in-process testing, characterization testing. The testing may be controlled performed by quality control, process development, or in some cases the client. Now, process development testing can be done, but it's only to increase process knowledge, for example, characterization. We would not use process development testing to make a decision about batch disposition. That would need to come from QC through qualified processes. The plan should be established before transfer to manufacturing. And again, that may seem early to some folks, but it really does facilitate the QC assay qualification and the appropriate matrices. So if we know where the sample is being taken, then QC knows where to do the qualification and which matrices to use for the qualification of the assays. Also, samples and specifications are integrated into multiple documents, so again, it's important to establish those early on. Plus, if we have samples that have to be shipped out either to the client or potentially to an outsource or to another site, although most assays are done internally, in some cases the client prefers to do some assays, or there may be a limited number of assays that might be done off-site. It's important to get those logistics worked out ahead of time. So again, another reason to have the sample plan established early on. Of course, we can accommodate late and ad hoc sample requests, but it's logistically more difficult, so it's more advisable to work through those sample issues early on. The other area is materials. It's important that the company, the client, identify any company-specific requirements. If there's any policies of certain vendors or grades of material or so forth, it's good to know that ahead of time. We will want to use the same raw materials, the same vendor and grade of raw materials used in development and GMP manufacturing, so that's why we'd like to know that early on. Of course, Catalent maintains an extensive catalog of standard raw materials that are used in GMP manufacturing, but if there are any client-specific raw material or specifications, those specifications would be reviewed and approved by the client. There may also be a release test method that may also need to be developed and qualified for that particular raw material. And the bill of materials is used in GMP manufacturing is approved by the client, so we have clear understanding of what materials will be used in the manufacturing process. So really to wrap up, just a few key points here. Again, project success depends on shared understanding of objectives. The client should consider their desired extent of involvement during process development. Clients should understand resource requirements and plan accordingly, and this is particularly important to understand the significant increase in resource demand, including client quality during batch record development, and also the potential need for real-time decision or at least concurrence with decisions during the manufacturing process. Testing and material strategy should be considered early on. And again, best results are achieved when the client and CDMO engage in a collaborative relationship, because ultimately both parties are working together to make therapies available to patients. As we say here at Catalent, patient first. So that's the conclusion of my talk. I'd like to thank you for listening and see if there are any questions.
Okay, great, Steve. So the first question is, at what stage is process development typically performed, and is there a time when you can no longer go back and do additional process development?
Yeah, it's a very good question. Typically, again, the process may come to us already developed, and we may just be refining the process or doing a facility fit adaptation, or we may develop the process completely ourselves from just from the initial clone of the cells. It may be possible, typically there might be an early phase process, and then when we go into later stages, phase 3, as I had mentioned before, it may make sense to do some additional optimization of the process. Typically after phase 3, though, the process is fairly well fixed. That's going to be done. You're going to be doing you'll be doing pivotal trials at that point, fairly large trials, and so it'll be more difficult to change the process at that point. You may need to do, if there is a need for a process change, they may need to do comparability protocols and so forth. As we get further on in our change control processes also attached as well, so it becomes more difficult to make changes after phase 3. So I think typically there might be an initial process for early phase that may be optimized going into phase 3, but after phase 3, it's fairly well established, and any further optimization would involve change controls and comparability protocols, and probably more involved regulatory.
Okay, great. Thanks, Steve.
Thank you, Leah. It was a pleasure to do the webcast, and thank you very much for this opportunity.
Great. And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website. As a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.